And when I thought about what to present as part of the INI program, I went through the existing program and I didn't see much about delays, and so I thought that might be a nice change. So today I will talk about the use of delays in modeling and simulation of biochemical reaction systems, and also on using those delays for exact model reduction. So let me start by introducing mathematical models with delays. So I tried to trace back the history to the beginning of the use of delay differential equations, and the earliest I could find, and that does not mean that there was something earlier, that was Volterra. So Volterra used delay differential equations. In fact, he was even interested in not only constant delays but also distributed delays already at his time, and that was likely in the early 20th century. Since then, delays have become more and more popular, and in particular for describing biological and physiological processes, and I give you a few examples. For instance, so here we have a viral dynamic model. Specifically, it's HIV, so the virus enters the cell. Then we have an intracellular time delay until viral production and the release. There are delay models for tumor growth, for drug therapy, then immune response. Obviously, there's some time associated with the immune system realizing that there is an intruder. Respiratory systems, here we see a recent model from 2016. So obviously there is a delay involved in the transport of oxygen to the tissue and of CO2. And then lastly, organism development. So here we see an embryo of a zebrafish, and the somites that have formed here, and those are formed in a 30-minute cycle, starting from the tip, and there's the presomitic mesoderm here. So the tail goes in this direction, and this has been modeled also using delay differential equations. But there are many, many more. Now, why do we use delays? So obviously they provide a mechanism to model processes without adding too much delay. These processes are quite complex, and we just lump the mechanisms and the intermediate steps that we either do not want to explicitly model or that we cannot model because we don't have enough knowledge. And then, of course, the time that such a process requires is included in the model as a delay. And so this allows many times for a more intuitive modeling approach. So the modeling is then more physiological, not mechanistic. But the approach is particularly useful if we have information about the delay, either derived from observations or from theory. So in the first part, I will talk about stochastic simulations with delay. I wanted to start with the chemical master equation, but we have seen that now so many times in the last weeks that I will just skip this, and I will go straight to delay stochastic simulation algorithms. So some early work, and it's basically 10 years old already, by Brazzo published in PNAS, and by Mario Barrett, myself, and Kian in PLOS Computational Biology. This work introduced delays into the SSA. And since then, various versions of such an SSA with delays have been published. The differences between those two versions were in the way they deal with the waiting time and introducing delayed consuming and non-consuming reactions. That's what we did in our publication. and with the time steps in the presence of delayed reaction updates. Slightly newer implementations are here by Roussel and Su, 2006, Kai, 2007. That was coined the exact DSSA, as he really calculates the exact probability for the next reaction to happen in any of the time intervals that the update points of delays establish. And then, of course, there is David Anderson's algorithm from 2007, very nice work, where he also, he presents the modified next reaction method and includes not only delays but also time varying rates. So that's the algorithm, what was later coined the rejection method by Kai. It's exact, but it's very simple to implement, and this is why I actually present it. We have the classical SSA, and whenever we have a delay, then we have to, of course, memorize this for the future, that there will be an update point. And whenever we pass this update point, then we have to reject the method, the reaction that we drew, and then just go to the update point of the delayed reaction, and from there we continue. Now, there is something worth mentioning about the difference between the consuming and non-consuming delayed reactions. So in the PNAS 2005 paper, Bratsun studied delays in protein degradation, and they claimed that this can cause oscillatory behavior. Later on in 2011, Miekic and others showed in their paper in the Journal of Mathematical Biology that in fact this is just a consequence of regarding the degradation as a non-consuming reaction. And here I printed a solution of a very simple system where we have the DNA goes to a protein directly. Protein is degraded, non-delayed constitutively, but also either with a delayed consuming degradation or a delayed non-consuming degradation. And in the case of the delayed non-consuming degradation, you see some kind of oscillatory behavior. And so depending on how you use the delay, you can get very different behaviors. In 2006, we applied our DSSA algorithm to the following example. So that's HES1, that's a delta notch signaling molecule. So HES1 is a negative autoregulator. Previously, it had been reported from experiments in mice that HES1 mRNA and HES1 protein levels oscillate with a 2-hour cycle. In their publication in Science, Hirata et al. Also introduced a model. For that, they needed a certain species to obtain the oscillations, but obviously there was no experimental evidence. But then later on, it was Nick Monk and others that showed that if you introduce delay in the transcription or the lung transcription translation process, then you actually get nice oscillations. So we reproduced this kind of this work. We used this model, but we used the DSSA, and of course we also got nice oscillations, but for a wider parameter space. So parameters here are, for instance, the cooperativity age, so the Hill coefficient here, or factor, as well as the delay. Another nice result was the following. So the Western blots of HES1 protein from whole cell populations suggested that there was a damping in HES1 mRNA and protein levels. But our simulations suggested that this damping stems actually from an averaging over the whole cell population, but that it is more likely that on an individual cell level these oscillations continue, and this was later on also confirmed by single cell experiments. So then We also did some work on the delay chemical master equation that we introduced in this paper. Since our model only included delay non-consuming reactions, so our DCME had these four terms, so two here. This is here we introduce a delay, and this is a standard CME for all non-delayed reactions. Recently, we published in Proceedings of the Royal Society A paper on delay chemical master equations and when this equation can actually be solved. And we also introduced here the delayed consuming reactions. So here, obviously, you get additional four terms. And this is simply because we have two update points at the beginning when the reactants are consumed and at the time when the products are formed.
So can you clarify, so you only need to look at the values of the function at a discrete set of backwards times. You don't have to drag a whole interval of history with you?
So of course, just like in DDEs, you need to specify history. That's point one. The second is, so what we wrote down here is for a discrete time delay. But, and let me go to the next slide, you obviously can introduce distributed delays, and then you end up with these additional integral terms. I will present later examples for when this can actually be solved.
For the discrete delay case, do you have to drag around an interval of past information or just a finite set of past times? There's a whole interval.
No, you have to have basically the whole history covered, because you don't know in what time are you actually looking back in history. Looking back in time. So now let me talk briefly about exact model reduction, and for the moment I just look at temporal processes. So the motivation is, I think, rather obvious. So biological processes have many reactions, many chemical species. The chemical reactions are discrete stochastic events, so continuous and deterministic approaches may not be accurate. Obviously, if we look at the CME, then solving that can be, or will be in most cases, impossible. Of course, we can use stochastic simulation algorithms, but even those can be prohibitively expensive for large numbers of molecules and reactions. So the goal in the reduction of chemical reaction networks is to gain significant computational savings by reducing the model. So we construct a simpler reduced reaction network that accurately describes the dynamics of one or more species of interest from the original system. So typically, this means that the abridged system has fewer chemical species or fewer reactions or fewer states. So the state space is reduced. Here I give you an overview of reduction methods. This is by no means complete. One of the earliest is actually the work by Michaelis and Menton, the equilibrium approximation. Then a few years later, Briggs and Haldane they introduced the quasi-steady state approximation. Then this was extended with singular perturbation theory, where they remove highly reactive intermediates of low numbers, as in the paper by Masny. There is one reaction abridgment. Here in this paper by Gillespie, they look at A going to B and going back to A, but also B going to C, and when can this be abridged by A going to C? Then we have the finite state projection by Munsky and Kamash, so this reduces the state space. And lastly, the paper by Thomas et al. From 2012, the slow scale linear noise approximation, where they use a reduced linear larger than equation that still accurately describes fluctuations in slowly varying species. So we propose here a different model reduction technique. The idea is to understand the changes basically as a random walk. So we start on the left, this is our red, and it sees the cheese, and it takes some time, and voila, there it is. It changed its fur color, but it also has gained weight. And well, what we want to do is, instead of simulating the individual steps, we are just interested in the first and the last state, not in whatever happens on the way. So we associate a delay with this process, and we obviously have to draw from a certain distribution. And then we use either a DSSA or also a delay differential equation, but then with not with a constant, but with a distributed delay to simulate or to solve the abridged system. Now, here you see again the original model and the abridgment. So species S0 becomes Sn plus 1. It goes out with a certain reaction rate C0, and then we have a delay associated. This is the time that for S1 to become actually the molecule Sn plus 1, and then the question is how long does it take?
Can I ask something? So, are you interested in how much time does it take for one molecule, basically? Is it just, I mean, because...
In this particular example, when it is linear, if you think of a random walk, then the walkers do not interact. So it's the same time for one or for the next or for the third molecule. They all follow the same distribution. That's why we only need to calculate one distribution. It gets way more complicated if you actually have nonlinear reactions on the way. And if I have time, I may go back to that later. So, yeah, so as I said, so how long does this take? And we obviously see here the equivalence to the first passage problem. So random walker starts in S1 and is absorbed in Sn plus 1. And with Tn plus 1, we denote the time until the walker arrives. And then I will quickly go through now the derivation of the PDF or the CDF. So the CDF of Tn plus 1 is described by this probability, starting at time 0 in S1, when do we arrive at Sn plus 1 at time t. Now it follows from the chemical master equation that what we actually need to look at is at this term, so Cn times p, so the probability of being in Sn at time t. Then we can obtain this term by solving this differential equation for where p of t is this vector. And we already see here that what we are actually interested in is the last component. A is the rate matrix of our system, and obviously we already know the solution, which is simply the matrix exponential, so E of AT times P naught. Now we can do a little bit better. So obviously we can just solve the matrix exponential using some numerical methods, but we actually can derive a closed form. For this, we look at the transition matrix A. It has a tridiagonal shape. We know the determinant. We can say something about the eigenvalues, namely that all eigenvalues are not zero with negative real parts using Gershwin's circle theory. And moreover, it's similar to a Hermitian matrix because all eigenvalues lambda k are real and simple. We will use all of that later in the derivation. What we use first is the Laplace transform. So the matrix exponential can be written in this form. This is called the resolvent. As I said, we only need element n1 of this P of S. For that we can apply Cramer's rule. Then this looks like this, where we have the minor 1, n of matrix A. Now I omit additional steps, but finally we get the following solution. So the Cn times Pn of S that we are interested in is Cn times minus 1n plus 1 times this term, and knowing the determinant and knowing something about the minor lets us plug in this fraction, and then we can do more math, and eventually we end up with this product form. And then we apply the inverse Laplace, and obviously, or rather straightforwardly, we obtain that our solution, so the PDF, is a convolution of exponential distributions with parameters minus lambda k's. Since the lambda k are all negative, so these minus lambda k are positive, and we can even get from there the CDF, so there is a closed form, assuming that all the eigenvalues are distinct, then this looks like this function. If they are not distinct, then the function is way more complicated, and but there is still a closed solution.
You're saying it's equivalent to actually having like an n steps where there's just some exponential delay at each step, even though it's a birth-death chain and you go up or down.
Right. I mean, if you have just one step, then the whole thing reduces just to the exponential.
Sure. So here you know, just all you have to do is you have to compute these lambdas rather than read off the constant, right?
Right. I just give you two examples. So here we have a chain of linear reactions of length 8. We use those parameters, and then we nicely see that we get a very good agreement with the SSA solution using the DSSA, so in green and in red. Now what we also played around with is how many of these lambdas do we actually need. And it actually turns out that if we just use the first eigenvalue, then I mean, it might not be as perfect, but it's a pretty good approximation to the solution. So especially if we have very large matrices, then it might not be necessary to get all the eigenvalues, which is a problem on its own.
So if you have a single one, it's your back to Markovian model, right?
Right.
Yeah. This will be.
This is another example. Here we try to get an idea of the average speed up of this rather small system. So S0 becomes S1, S1 becomes S2, and backwards. And lastly, S2 can turn into S3. And we just assume here that the forward and the backward reaction are identical. And then for different parameter values for C1/C1R and C2, we get different eigenvalues. And also we see here what is the average number of SSA reactions per single DSSA reaction. And obviously this is correlated with the speed up of the system. So if the difference, let's say between the C1 or C1R and C2 is huge, then we obtain also the biggest, the largest speed up. Now we can extend this by introducing, well, what we call bypass reactions. We can also introduce degradation as well as incoming reactions. And this all works. I just give you again some examples. So this is a backward bypass reaction, just a single one. And again, there is very nice agreement with the SSA. Here is a forward bypass reaction. You can also mix forward, backward, etc. as you wish. Now, if you introduce additional degradation reactions, then you actually have to modify the DSSA at least a little bit. So first of all, we need then an absorbing state as not, and all degradation reactions go then towards this absorbing state. Then the first passage time is described by actually a pseudo-distribution function. But for t going to infinity, this pseudo-CDF converges to a cumulative limit, W or omega. This will be smaller than 1, and 1 minus omega is then the degradation probability. And in the DSSA, we have to make the choice, will our, let's say, Walker or will our protein, will it degrade, or will it reach the final state? And when we know that it reaches the final state, we can draw from the delay distribution and continue simulation. So again, the degradation also works nicely. Again, it matches both the histogram for a certain time point as well as the time evolution. Now we can look at nonlinear reactions. So how does it work for a Michaelis-Menten reaction? And obviously we cannot expect that this is exact. However, under certain circumstances, we can abridge this system with the following. Yeah, so S plus E becomes now the product plus the enzyme. And we have to introduce here a new reaction rate K and delay. And obviously the question is, well, how do we choose K? And since this is an artificial K, what we can do is we can introduce a very, very high reaction rate such that the waiting time for the reaction to happen is much smaller than the delay. And so this is the first approximation. And then the next question is, how do we choose the delay? So with one delay distribution, I can already say an exact lumping is not possible, as in principle each state requires a different delay distribution. Now we still applied it to Michaelis-Menten, where we either have much more substrate or much more enzymes, and in this case what we can do is we can just linearize the whole thing. So we use a larger initial condition as a factor in the corresponding coefficient in the transition matrix A, and then this yields a constant delay distribution and a good approximation in these cases. So here I show you one example, and on the left for species S and on the left for species P. And the difference here, I believe, is 2 to 3 orders of magnitude difference between the enzyme and the substrate concentration at the initial condition. Perhaps a more meaningful example, here we model a chain of reactions in mRNA decay. This is from a paper in RNA from 2001 by Cowan and Parker. And so what we abridge here is this part that you can probably perhaps not see very well. So what we say is, well, we are interested in fragment I2, okay, and so we abridge the whole system with in the following way. So our mRNA is degraded and split into different components and fragments, but with I2 it will be with a certain time delay tau, and we of course also need the probability that it gets degraded or becomes a different fragment. And well, with this parameter set we simulated this both in the SSA and with the DSSA, and again these things match nicely. Now we can extend this further to fully monomolecular systems. You probably noticed in the schemes that I showed you earlier, we always had the reaction just going out but not going backwards. Now if we want to abridge this, then we have to account for the possibility that a molecule actually goes back to its original state. So we do not only deal now with a first let's say passage time or first arrival time, but also with the first return time. And this is illustrated here for S1. So there is a time associated with an S1 molecule becoming S2 and whatever else, but going back to S1 before it goes to Sn. Now in this case we even have actually a closed loop. So it's basically all possible. And we can even abridge this if we are only interested, so if our species of interest is only S1, then we can just approach this with a single delay distribution, just like this, and it's fully exact. Now, I mentioned already that we have to do a little bit more. We need additional distributions, but we also need the probability for a walker that starts in S1 to return to S1, or starts Sn to return to Sn, and we do not only need 2 return times, but also now 2 arrival times, depending on whether you start on S1 or Sn. Then this is these are the return probabilities. This follows basically the same train of thought as outlined earlier. We basically can calculate this numerically using the matrix exponentials, but we can also obtain closed forms. Now, this is for the first arrival. So here we have the following system, and it turns out to be again the convolution of exponential distributions with those negative eigenvalues, and this is simply the matrix shape. Again, it's diagonal, so it all works well what we applied earlier. And this is actually rather counterintuitive. The first arrival at S1 time distribution is identical to the first arrival at Sn time distribution. And it's not immediately clear why this is so, because one could think that if you have, let's say, if all the reactions that go to the right are much stronger or much faster than those that go in the other direction, then the distribution would probably change. But what changes is only the probability to go from one to the other, but not the distribution. Now the sketch of the proof is outlined here. So we just rename Sn, let's say, with S1 prime and Sn minus 1 with S2 prime, etc. Then the system changes. It looks like this. We have now a matrix A prime. The first arrival at S1 becomes now the first arrival at Sn prime. But since A prime and A are similar, the eigenvalues have to be identical, as those are invariant under similarity transformations. And we have done several simulations or calculations of the distributions, and indeed they are identical. So we can also derive a form for the first return distribution. I will not go into details, but as you can see, it becomes more and more complicated. Since we do not need to solve a matrix exponential, we have the closed form. We can immediately draw from this distribution. This also yields some computational savings. I give you here another example. This we took from a paper by McKeeson from 1995. So this is a reaction scheme for T-cell kinetic proofreading, where you have this long chain of unimolecular reactions. Our reduced model looks like this, where P is the probability that actually our system ends up in C6. So that's our together with T and M, these are the species of interest. We can get rid of all the other species and of all the other reactions, but we need the probability of ending up at C6 and for returning without reaching C6. Well, this we have here. And then we have now 2 delay distributions, namely the arrival distribution to go and reach state C6, and the return distribution for going back to the original state. And again, as you can see nicely, the dynamics match very well, and this is how the 2 distributions first arrival and the first return distribution look like. So what are the benefits of this approach? So first of all, the lumping is exact. Yeah, we can do this, as I've shown, for linear reaction chains, including degradation, synthesis, and those bypass reactions. It's independent of reaction rates and initial number of molecules. So there is no need for any timescale separation. It works within the existing DSSA framework, and the nice thing is that it's reusable, so we can think of modules, or we can think of a linear chain that we replace by a delayed reaction in any other non-delayed one. Because otherwise, if we only look at the linear chain, then obviously we know already the solution of the chemical master equation, which is simply a convolution of multinomial distributions with a Poisson distribution, which has been shown some time ago by Huizinga and Janke. Now, in part 3, I briefly extend this to spatiotemporal processes. This is work that Kevin, Tatjana, and I did some years ago, and it appeared in BMC Systems Biology. So the idea is here that we obtain the delay distribution, perhaps from single cell experiments. So somehow we have information about the diffusive profile. In our case, since we did not have such data, we obtained it from a particle simulator. We used at that time ChemCell. And from there, you again get a distribution. So in this case, let's say for a transport process from a cluster somewhere near the membrane to the nucleus. Once we have this distribution, we can just model this via a simple reaction AC goes to AN with a certain delay. And as you can see here, since the solutions are really on top of each other, you cannot see very well, but I can tell you that the DSSA solution and the spatial the simulation results are absolutely identical. We then extended this, we incorporate different other types of reactions, either with reaction rates or by including delays, even for binary reactions, like in scenario 4 here. But we also have even more complicated examples now. The whole thing is no longer exact because we have nonlinear reactions, but the error is still rather small. So for instance, for scenario 5, we can go back. So this is the full scenario here. We have translocation, we have a binary reaction, and we have diffusion out of the nucleus. So for this example, for instance, we had we ran ChemCell. It took us 15 minutes, and using the DSSA, after obtaining the distribution, it took us only 0.3 seconds to simulate this. Now, this is another interesting story. So what we do here is also for scenario 5, but what we do here is we assume we have the spatial data given, and then we either try to find an ODE that matches it, or a DDE with a constant delay, or we use our DSSA. So we do a parameter fitting of the reaction rates in the ODE model, given the spatial data, and this actually does rather poorly. So this is a total relative error, and that's basically the relative error of all these 5 species that are involved with respect to ChemCell simulations. And we can do the same now using a DDE. Here we fitted a constant delay, and obviously it's already better. But if we have the delay distribution and we plug it into the DSSA, then the error is much smaller. Now I have, I don't know, maybe 5 to 10 minutes left. So let me quickly talk about a very recent work that we did. We were interested in examples when the delay chemical mass equation can be solved exactly. And we found 2 examples. Obviously, one is a very simple linear unidirectional reaction system that we will average like this. And another example that I will give you is this work, this model. We used the model from a paper by Platini and others in PRE in 2013 on mRNA maturation. And this is then the abridgment. So the mRNA, the maturation of the mRNA is lumped in one delayed step. And so here I show you how the DSCME actually looks like for the first example. So here X is just this tuple, so X1 and X2, which describes the number of molecules in state 0 and state 4 respectively. And we have again these very nasty terms. And of course what is really bothering here is A, the sum, right, over all previous states. So Xi is a state in the past at time t minus s from which the system evolved to enter state x at time t. Okay, and of course also these probabilities here are not so easy to simplify. However, for that particular system, we could find a solution just by looking at probabilities. So here are the arguments. So what we know is that if we have, let's say again, our walker in state 0, then we can write down the time until it takes to reach state 4. And this is described here by what we call t for 0 of t. This is the CDF of the first passage time. Then we also have a probability for staying in state 0. That's 1 minus f of t. So f of t is basically the CDF of this exponential probability density. And then the third state that we have is that actually our walker is not in state 0 and not in state 4, but somewhere on the way, which is simply then 1 minus those two. And we can simplify this to f of t minus t40. But then we basically have 3 event probabilities, right? The walker can be in state 0, the walker can be in state 4, and the walker can be on the way. And it turns out that the solution is nothing else than this multinomial. And again, this reminds us of the paper by Huizinga and Jahnke, where they expressed the solution in terms of a convolution of multinomial and Poisson distributions. But in this case, we use actually a delay. So the delay, the distribution is included here in the CDFs of the first passage time. Now, for the second example, we actually needed to use a different technique. Here again, you have the DCME. Well, the first terms here, they look nice, but again, the question is what can we make out of these summed integral terms? And as I can show you now on my second last slide, we can indeed do a simplification. So forget about the integrals for a moment. If we assume we have a constant delay, then we can basically, in this case, write this as in product form, and this reduces further to this term. And well, we have to do this twice, right? When leaving a certain state as well as for those terms where we describe the probability of entering a state, we can do the same, and then we get this. So we can replace then those terms in our delay chemical master equation and get this much nicer form, which we can then also solve numerically. Now the simplification, and that's the important point, is here the simplification is possible because the triggering of the delay distribution is fully independent of the occurrences of other reactions or of the state at the time of triggering. Because the DNA is, in our model, it's not stating, we just have one DNA. And so the propensity is constant, it doesn't change, and only in this case we can actually split these joint probability terms. And well, with that, I'm at the end of my talk. So these are the collaborators in the project that I presented to you. Manuel Barrio from the University of Valladolid, Kevin, who is here, Tatiana, and Tian, who is now at the university, at the Monash University in Vienna. Thank you very much.
Yeah, I have two questions. I mean, you said at some point that this, I mean, you had this big formula with convolutions, and you said that this would be faster than calculating the metric exponential. But are you essentially, in order to have this formula, do you need to calculate the eigenvalues of the matrix?
That's right.
But I mean, isn't that more or less equivalent with calculating the matrix exponential?
Well, so for big systems, if you need all eigenvalues, then actually that can be quite time-consuming. If you have a smaller system, or if you are just interested in the main eigenvalue, then this can be faster.
Yeah.
So it depends a little bit. Also, of course, once you have the distribution, you plug this into the DSSA, so you know it's not necessary for those systems to recalculate the distributions. And I forgot to mention this. It's actually different if you introduce, for instance, bimolecular reactions, because then you have to calculate state-dependent delay distributions. So basically, on the go, when running the DSSA, you would have to calculate, well, from what distribution do I have to draw now?
Because it changes depending where you are.
Exactly, and this basically destroys the whole method, because you will never be faster than just running an SSA.
And then the second question, which is, I guess, perhaps a very naive one. So, when you wrote down the simple chain, I mean, this has a well-defined kind of invariant distribution. Does it make sense to talk about invariant distributions in a delayed system, or the system never reaches equilibrium, strictly speaking?
That's a good question.
I think you can, and I think Ruth Williams had a paper with someone called Kelly, who was also a PhD student. Ruth Williams.
Well, maybe we have to look into that one.
Okay. Thank you.
Thanks.
Another naive question. So if you had, instead of a continuous time Markov chain, is a discrete time Markov chain, or a fixed delay, fixed time for each step to occur, would your calculation technique still work? Would you get some closed form expression for the probability distribution, or the number of steps it takes to get from 0 to n?
Well, so we derived the delay distribution to be exact, right? Because it's not a constant delay.
Sorry, what I'm saying is, in other words, I guess what I'm saying is, if instead of calculating the probability distribution for the time it takes to get from state 1 to state n, could you calculate the probability distribution of the number of transitions it takes to get from state 1 to state n?
Probably, yeah. I mean, I think it should be possible. Yeah.
What was the point?
I'm just curious whether there's a discrete time Markov chain.
Okay.
You guess, but who knows? Maybe exponential makes it nicer.
I guess one would just have to write it down, but.
So you had your spatial model with binary reactions. So what exactly do you do? When you have two molecules that react, you have to compute an average delay, because I guess it depends on their position. Was they are initially, or was they in
Right.
space?
So I showed one example of Michaelis-Menten, and in this case, if one species is in excess, then
Okay.
what we did in this case was we just linearize it as if it is just an
So they
addition
are
to the
everywhere,
constant.
and it's only dependent on one species.
Right.
Okay.
Now, if you want to be more exact, then you actually can do this, and we have done some work on this. We just have not published it yet because there are no computational savings. So it's interesting from a theoretical point of view, but it does not have a practical value because the delay distributions are state dependent. So in fact, you would have to still monitor the state of species that you actually want to get rid of. I mean, you can do this, so you can still transform everything into delays, but there are no savings. That's essentially the.
Even if you pre-compute the delay, or
Even
figure
if
out
you,
some
that's right.
structural-
We also tried that. We pre-compute this. One rather recent idea I had was, well, maybe we do not need to follow up on every single update or change in the species of interest, right? So we could say, well, maybe it's enough if we know the time delay until we have 10 more proteins instead of one and one. So one could perhaps look into this. So that would be some kind of tau leaping with delays, right? I mean, we once actually, so we also have work where we extend the tau leaping algorithm with delays, so that also works. But if how this now fits to this framework, one would have to see.
When you say tau leaping, is it equivalent to saying that you're coarse graining the space, and instead of computing your delay for every vertex in the state space, you compute it for every region?
So tau leaping with delay is you just have delayed reactions, but you still try to choose the time step long enough such that a certain number of reactions happens in which the propensity does not change too much. Well, the delays, because you can have some reactions can be delayed. You just have to trigger those, and you still have to keep in mind when to update. So it's just tau leaping basically.
Any other questions? Okay, let's thank the speaker again.